Hello friends, welcome to the first part of the second lecture of week 12. So in the previous lecture we discussed what is metagenomics, examples of metagenomes, what we do in metagenomics and what is the outcome of it. So in this part we will look at the sequencing and assembly of metagenomes, their function annotation, including gene prediction and annotation of those genes, right, and construction of MAGs. So we will divide this lecture into two parts. So metagenomics is the science to sequence the unculturable community. So basically whatever is their representation in that environment, we take the sample from that environment and sequence each and every prokaryote that is there, the bacterial cells, right, without actually culturing them. So this can basically tell us, we also saw in the last lecture, that what microbes are there, how many are there, what are they doing in that system, and how they are performing those functions, and for which we have to perform the sequencing, which can be long read and short read. So basically metagenomics, there can be two strategies. One is just to know that what is there, so that we can do by the 16S ribosomal RNA, which is a gene present in all the bacteria, and it is conserved as well as variable. So it contains conserved, constant and variable regions, so which can be very helpful. For example, V3 is one of the variable region in 16S. So either we can amplify the complete 16S using primers and make a library of it, okay, or go for direct sequencing using long read or combination of short reads, or we can go for any selected hypervariable region and then identify the bacterial species, because these are markers. So there is a database that is available like reference RDP or green genes, so we can map against that and know that what species are there, okay. Or basically species information may not be that much, but at least genus level we can say that what all is present in your sample. Other is metagenomic approach, where we, using the shotgun fragmentation method, we sequence all the bacteria that is there, make like small pieces of that, okay, and then go for the analysis, right. So analysis include like ORF prediction or gene prediction, function annotation. So basically those ORFs which have got a function may be called as genes, right, and then the metabolic reconstruction to see that what all pathways or what are the particular features of that microbiome or particular MAGs or species which are abundant in those, right. So amplicon based approach is targeted or 16S based approach, right, and WGS approach is basically you want to cover the entire genome. So basically in this case you are dealing with about 1500 base pair or 1.5 kb gene per bacteria, but here you are dealing with the complete bacterial genome, that is like 3 to 5 MB or like that, okay. So it will be much more to sequence, and basically this can be easily done in like 200 300 MB, million base pairs total data that you get from sequencing, and for this one you need to have, for metagenomic you need to have at least 4 to 10 GB or even more from particular sample. Okay, so basically the strategy for this is just a recap. You will collect the sample, clearly, like this is for the fecal sample collection, transport under the conditions so that it does not change the microbiome of the sample, right, preferably like at least 4 degree or basically -20 is even better, right. Transport as early as possible in the lab or store in -20 immediately, and go for the extraction of metagenomic DNA using kits or your protocol, right, and then go for sequencing. Okay, so basically sequencing then again in this case can be short read or long read. So far short reads have predominated because bacterial genomes are short, and basically short reads can correctly cover, these are correct reads, basically they can cover the genomes, okay, and these are cheaper to produce also, right. Whereas long reads are now becoming popular because in a combination of short reads, of course, because they can cover this entire 16S ribosomal RNA or complete genes, right, in one go, which you will take a lot of time in assembly when you are using the short reads. Okay, so NextSeq 500 is one of the sequencer which can be used, or basically MySeq or MiniSeq can also be used for this purpose. And they are good because they are low budget sequencers which you can afford in your labs, and basically they can produce the required amount of metagenomic data. Okay, and yes, basically multiplexing and barcoding is one of the feature of these, because if you want to populate say 40 GB minimal that this sequencer can produce, right, and per GB you want say 10 GB, so basically you can put 4 samples in this, right. So basically 4 samples with different barcodes you can put, okay, and later on you can demultiplex those barcodes, so you don't have to waste the your remaining space if you have just so you can use multiple samples together, right. So and for the metagenome, it could be that the amount of DNA that you extract may be less, right, because you don't want to go for PCR amplification to get at any bias. So you can use NextEra kits which can work with even 1 nanogram, okay, or okay, NextEra Flex which can 1 to 500 nanograms. So basically these can be used for low amount of DNA, and TrueSeq kits we generally use for larger genomes because they require more amount of DNA. Okay, TrueSeq Nano still is there, which may be cheaper than NextEra, right? And similarly, the other bioanalyzer kits and the qubit high sensitivity. So those kits are also according to the metagenomic libraries that you want to prepare, right? So and then long reads can be produced by so many ONT or packed biosequencer. For example, the ONT ones may be cheaper and more affordable for you. So like this is their cheapest, it comes free with the consumable, actually this sequencer, and this is USB based sequencer, and you need a decent laptop with about 1 TB disk space, and this can do the job, right. Or this MinION, or basically these bigger ones are for like larger centers, but these two are more preferable for shorter smaller groups, right. Basically from the sequences that you have got, so you can see sequences with multiple color, they are coming from different genomes. So first of all, there can be several approaches. One is like marker based approach, where you compare these sequences against a marker database and identify the species, and based on their abundance and presence you will build the phylogenetic or taxonomic tree, okay. Or you can do binning of these into different group, red color, green color, blue color, which may indicate that what genome they might be coming from, okay. And then you can directly calculate their taxonomic abundance for each of those bins and their representation or relative abundance, right. Or then again with against a reference database and build the tree, okay. Or directly against the genomes and see that what genes or pathways they might be having, right. Or you can also try to attempt an assembly, okay. If you think that for some genomes you may have a higher relative abundance, or at least even by combining multiple samples you can recover the complete genomes from that. Okay, so basically in the last lecture we will look at the 16S amplicon analysis or taxonomic abundance and some case studies. So basically it starts with sequencing. The data is demultiplexed because you might have pooled multiple samples together using multiple barcodes, and then you also have to got rid of the adapters and those barcodes from these sequences, right. Then if the insert size was smaller, okay, and forward and reverse read have some overlap, okay, so you can create these into a single read, okay. This could be 150 base pair, this could be 150 base pair, overlap of the 10 base pair due to the insert size, and you may get like something like 290 base pair or something, okay, longer read. Or you can go for direct assembly, right, and then identify the contigs, right, for the marker, right. So basically assembly will only be done when you are dealing with paired and read which have some overlap, okay. Otherwise you will directly go for the constructing the OTU table, right, and followed by taxonomic classification and various statistical analysis that we will also see in the next lecture, okay. And for the metagenomes, basically again either you generate your own data or take from publicly available database and do the quality filtering using fast QC and Turbomatic. Okay, that also we have discussed so many times in case of prokaryotic and eukaryotic genomes, right? You have to get rid of the human contamination or any environmental contamination that may happen, because for example, human host, if you are deriving something, say human gut microbiome or skin microbiome, there could be contamination by the human cell or human fecal DNA. So for that you have to use BM Tiger or Botai2 to remove the components of eukaryotes or humans from that data, because otherwise your analysis will be biased and not be correct. Okay. Then you will go for taxonomic assignment using Metafilin 4 or Kraken 2 or Kaiju. All three are like standard and good tools, right? And then prepare the abundance table, that which genomes are there, what is the relative abundance, right? And then see in sample to sample or healthy disease or whatever your question may be, that whether how the abundance is varying in the two environments, and supported by the statistics, right? And then the downstream analysis, construction of figures and these things, right? So for the assembly, one of the most used tool is Metaspades, okay, which is very good metagenomic assembler, okay, and it can assemble, it can do things fast, and basically it can assemble a large amount of data also, right? So basically from different sample you can see that from de novo assembly it is generating multiple contigs followed by the functional prediction and the functional gene composition. So basically Metaspades, so when you say you can download from GitHub, okay, right? So the if you say Metaspades py -- help, so it will display all the options which you can use while running this software, okay. So like -1, -2 could be forward read, reverse read, then paired and read, okay, output file name, number of threads or CPU event you want to specify, memory you want to allow this to take, right? So you can fine tune this. So for example, this is one of the standard command line. So metaspades.py -1 is the forward file, forward reads file, reverse read file. In among the paired and read you have forward and reverse reads, right? So forward read file, pair, when both the files have equal number of reads arranged in the same order, 1 2 3 4 5 6 and 1 2 3 5 6 in both forward and reverse, right? One forward, one reverse, one forward, one reverse in both the files, and both file will be like having equal amount number of reads. Number of threads you can specify that. Again, this could be if your system allows say 64 threads, so you can go for like 60 or 55 or 58, so that you allow some threads for other jobs to continue, okay, the basic jobs, and you can use maximize the thread usage. K-mer size, okay, and thread offer 33, the quality values, right, and the output folder name, okay. Then once your assembly is done, you have to do the like statistics or parameter, like what is the quality of your assembly. So for this you can use quast py, okay, command. So -o, basically that is your and this is your folder that will contain the quality values, okay, and this is the folder for basically at this point I've mentioned it like quast K21, because you might be using different K-mers for running. So at 21 this is the quality, 22 or 31 this is the quality. So basically this folder name quast K21 will be storing values for the quality or statistics for using K-mer 21. When you use another K-mer you can specify like quast 31, so that you don't get confused. So basically in bioinformatics it is very important to make the folders and organize the data cleanly and clearly so that you don't get confused. Okay, then the folder in which you generated the output out/contigs FASTA, okay. So out folder in which there is a file containing name contig.fasta, okay, which is the default file of this program output, right, and number of threads to calculate the quality, and this will give the statistics for contigs.fasta file in quast_21 K21 folder. Okay, so once your assembly is done, you have satisfied with the assembly quality by running multiple k-mers, and okay, so N50 is high, okay, and right, so you think that you have used most of the data in the assembly, right? So then you will go for the gene prediction, in which you will because see, in bacteria almost 80 to 90% of the total genome, so like if this is a bacterial genome, almost 80 to 90% of this will be genes, right, and remaining is intergenic region. So basically that is why gene prediction is easier and better in bacterial genomes, right? So you can use several gene prediction tools, okay. So at the start you may like to call them as ORF, not genes, because once you find a function for it, or you find it conserved across so many genomes, you may call it a conserved ORF, or then assign a function, or basically function gene you should call when you are sure that that ORF may have a functional annotation also. So you will run it on the contigs, assembled contigs, or you can run it on the raw reads also, because your raw read is at least 150 base pair, okay. So 150 base pair when you divide by 3 is 50 amino acids, okay. So that means you can find a partial gene, okay, present in that contig, okay. So that contig may contain the complete gene or partially, okay. So basically you can also go for direct contig read level gene prediction or contig level gene prediction, right, and then you use that information for the function annotation or building the pathways, etc., and getting more details about the community. So basically tools could be Metagenomic or Glimmer or Prodigal, right, which can be used for the gene prediction. So Prodigal is generally used these days. Okay, then the genes, again, like in case of eukaryotes, similar here also, it could be evidence based, okay, where you will be aligning against the known genomes and seeing that your fragment or contig is matching with that with sufficient identity similarity coverage, and if that is true, you will assign the same gene name or coordinates to this one, right? Okay, so other method is ab initio gene prediction, where you will look for gene like structures, right, that includes a good ORF length, okay, or the cut above the cutoff ORF length, or even partial genes you will consider because it's metagenomic, okay, possible RBS, okay, correct start codon if RBS is also there. So these kind of features that we'll also see in the later slides. So metagenomic gene prediction is little challenging, because here in the complete genomes you are dealing with complete genes that have a start and, okay, upstream region that includes RBS and Tata box and these things, right? But in metagenomic you are dealing with partial or incomplete gene, because that may come from any parts. For example, this is a genome, okay, and there are so many genes here, like this is one gene on the reverse strand, these are different genes. So in the read you may find that if the read is coming from, say, this region, okay, so it will contain some part of the 3' end of the yellow gene, okay, complete this gene, green gene, and partial of this blue gene, right? Similarly, if it is coming from this gene, so it will be having neither the start and not the end of this orange gene, so it will be partial. So how will you call these fragments of genes? So this is the challenge in this, okay. And then the copy number of genes could be an issue, because if you are dealing with partial, right, so some part could be here, some part could be here. So how to basically come to the conclusion that how many times that gene was present, because fragments could be too many, but overall when you combine the fragment length, okay, the number may be very different, okay. And then whether you should use the contigs which are assembled to predict the genes, so that you should do when you want to look for the complete genes, right? But because in contigs you have already lost the information about the number of reads that map to those contigs, right? So basically multiple type of strategies are used. So sometimes genes are predicted from contigs and then later map the reads on that to find the count of that gene, okay, that is one strategy, okay, or directly count the gene fragments in your reads, okay. So that is another complicacy here. And contamination from host, that could be like the genes, you think that it is coming from your microbial community, but it may be coming from the host or other micro eukaryotes that might be contaminating it. So Prodigal is one of the software which is fast and for accurate protein gene prediction in bacterial genomes and basically metagenomes. So it does work on finished genome, draft genome and metagenomes. Okay, for it is very fast, like for a E. coli genome 4 to 5 MB in size, it can do the gene prediction in only 10 seconds in a MacBook Pro. Okay, and it's using an unsupervised ML algorithm and doesn't need to be trained on any data, and it can learn on its own. Like Glimmer can also do this using hidden Markov models, HMMs, right? So it also considers the other like important properties such as RBS motifs, start codons, coding statistics, like how many times codons are coming. Okay, so that is why this has gained a lot of importance and use in recent times, and it can be downloaded from GitHub, right? So this is the paper that covered the Prodigal software. Okay, and now so many new versions have come for Prodigal, right? So for Prodigal, you can just say specify that Prodigal -i your genome file.fna, output file name and the protein file name. So basically A, O, okay, these things are explained in the next slide. Okay, and similarly for metagenome, right? So like this, Prodigal -a, okay, for the getting the translations or basically the protein file, translated gene file. Okay, -g for the what translation table to use. Okay, -d for the nucleotide sequences, right? And -o for output file. So basically you can provide several additional parameters to run Prodigal. Okay, next good gene tool is Metagenomic, which is okay, which is also used well, and you can also use it online or download and use it. So this is also widely used. So this is one of the options along with Prodigal that you can use. Okay, so like if you download it, then it will provide you again several options to predict. Okay, and the command line for this will be like, you can say gmhmmp, and then the file name, the output file name, output format name. Okay, whether you want output protein sequences also or only the nucleotide sequences. So you can say A, D, these kind of options you can define, and this can run this can do the prediction of proteins in this. So you can also use multiple of these. Okay, but Prodigal basically does the job well. Glimmer has been a gold standard for gene prediction in prokaryotes, and they have also come up with a software for prediction of genes in metagenomes. Okay, so it comes from Salzburg's lab, right? So basically Glimmer can also be used, but among the three, basically Prodigal I think is the most commonly used gene prediction software, which provides you the table that contains list of genes, also the gene sequence and nucleotide as well as protein you can always specify, right? And then the gene set is ready for further analysis. So basically in this lecture, which is the first part of this lecture, we covered the portions of sequencing, assembly of metagenomes and gene prediction, and in the second part we will look at the annotation and construction of mags in the second part. So this is good for now. Thank you.